Okay, thank you for the invitation and for being here. Let's take it back to 1900s, where Ivan Pavlov first discovered that the mere sight, smell, taste, and even the thought of a meal resulted in the secretion of gastric and pancreatic fluids in dogs. This was later named the cephalic phase of digestion, as it starts in the brain, or the cephalus, and was actually one of the first brilliant demonstrations of the fact that our brains and our guts are connected. This connection occurs in a bidirectional manner via various mechanisms, and recently the role of the gut bacteria, the trillions of microorganisms that are found in our colon, is said to play a critical role in this communication. Of course, the gut bacteria has various functions, the most important of which is to try to help us digest certain food components that we eat, such as dietary fiber, which is found in fruits, vegetables, legumes, and whole grain products. This occurs via a process called fermentation, and the outcome of which is the production of these molecules called short-chain fatty acids, namely acetate, propionate, and butyrate. These molecules are critically important for the health of our colon, and they are predominantly absorbed in that site. And the fraction that escapes absorption enters the systemic circulation, some of which can reach the brain and influence our psychological health. So over the past few years, we've been thinking about what are the mechanisms that mediate the relationship between the gut microbiota activity and brain function, as we know that the gut microbiota itself cannot travel to the brain to exert its influence due to the fact that under physiological condition, it is blocked by the blood-brain barrier and the gut barrier. And so I've been looking into short-chain fatty acids as one of those indirect pathways that could mediate the gut-brain relationship and the microbial activity. And I've been doing this in specific relation to the stress response in humans. So when we encounter a situation in life of an uncontrollable and unpredictable nature, an area in the brain called the hypothalamus sends an alarm system throughout the body that results in the production of the major stress hormone cortisol from the adrenal cortex. This hormone is critically important to help us fight or flee from the stressful situation that we have encountered. And different people react in different ways to one and the same stressors. And in prospective cohort studies, it's been shown that this can be predictive of later development of psychopathologies, gastrointestinal disorders, and cardiovascular disease. And so we thought that this would be an interesting outcome to try to understand the role of short-chain fatty acids in the gut-brain communication. So when we started this line of research, we wanted to do so in a mechanistic way, and we started with healthy subjects. And of course, we needed a way to induce stress in the lab to examine these relationships. And so we asked participants to place their hands in ice cold water and to perform mental arithmetics, all the while being videotaped and receiving really unempathic feedback from the researchers. So you can imagine that that would be pretty stressful. And indeed, we were able to induce a robust cortisol response that we would detect in saliva samples, accompanied by subjective negative emotions. And in addition to that, we would collect blood samples to quantify these short-chain fatty acids in the blood. So our aim was to try to understand whether the relationships that we see between the consumption of dietary fiber and mental health and psychobiological functioning could be mediated by short-chain fatty acids. And we wanted to manipulate or do our manipulation at the level of short-chain fatty acids directly in order to discover this relationship in a mechanistic way. And so what we have done is we actually took capsules and filled them with these very short-chain fatty acids, and we coated them in such a way that they would bypass the entire gastrointestinal tract until they reach the colon and release their content as if they have been fermented, as if the fiber that the participant would have eaten was fermented by the gut bacteria. And in comparison, we also administered placebo capsules that were also coated. So in this study, we invited 66 healthy male participants, and we divided them into 3 groups that came to the lab and underwent this stressful procedure. Afterwards, they were assigned to either consume placebo, a low or a high dose of short-chain fatty acids corresponding to 10 or 20 grams of dietary fiber every day for 7 days, and they came once again to the lab to undergo the stressful procedure again. And so what we have found first at the level of the short chain fatty acid concentrations in the blood is that prior to the intervention, the participants had comparable levels of acetate propionate butyrate, which did not change following the intake of placebo as expected. Whereas in the low and in the high dose short chain fatty acid groups, there was a significant increase and therefore uptake of these molecules into the systemic circulation. In relation to the cortisol response, prior to the intervention, the participants also had comparable levels of cortisol response to the acute stress, which also did not change following the placebo intervention. But in the low and the high dose groups, there was a marked decrease in the cortisol response to stress. So observing these patterns, we wondered whether short-chain fatty acids and cortisol would be associated with each other, and therefore give us an idea of whether the amount that is in the blood plays a role in this relationship. And this is indeed what we found. So those participants that had the lowest uptake of short-chain fatty acids in the blood tended to have the lowest decrease in cortisol response to stress. And those that took up most short-chain fatty acids therefore had the highest decrease in cortisol. At the level of the subjective negative emotions or stressful feelings, however, we did not see any change from before to after the intervention in our sample. And so we concluded that the colon delivery of short-chain fatty acids attenuated the cortisol response to stress, and that the magnitude of increase as a result of the delivery of the capsules into the systemic circulation may be a decisive factor. And so for the future, we are looking to understand the role of the individual short-chain fatty acids, which is one of our ongoing studies at the moment. And we're also looking to understand the mediating mechanisms. And finally, we want to know whether we would obtain the same results on the cortisol stress response upon the consumption of dietary fiber, and therefore whether that would indeed be mediated by the systemic concentrations of short-chain fatty acids. So if you're interested in the answer to the last question, make sure to come to my poster tomorrow. Thank you very much. And I would also like to thank my two groups, awesome people, the Laboratory of Digestion and Absorption and Brain Gut Access Studies at the KU Leuven for their help and support in this.